Hello, guys. I hope I started my recording. If I didn't, that wouldn't be good. So, if you don't mind, I would really want to make sure that I am recording. So, yes, I am recording. Excellent. The reason why I'm checking that is because a few times I did have a chance to talk for an hour, thinking that I'm recording myself, but I actually didn't press a button. So, to avoid that happen, I will have to check 2 times whether I pressed recording. Anyway, as you also, I hope you all saw the announcement, I decided to pre-record this lecture simply because in the place where I currently am, internet is so bad that when I had the last class, Bio 322, on Tuesday, I had horrible interruptions and students could not hear anything. And then I decided to go with pre-recorded lectures for all 3 classes, including that lecture that was interrupted on Tuesday. So basically 4 recordings. Anyway, I will get back in Edmonton on Tuesday, and then we will meet in person. I'm really looking forward to see you soon, especially for this class. My experience that I'm getting here will be extremely important. I learned so much, and this class will be literally through the semester. I will share with you everything what I learned here, and it will be a fantastic experience. So, let's start with the lecture that is on the schedule today, lecture 2 in unit 1, which is Introduction to Applied Microbiology. So as you know, last class we just briefly covered what we are going to do. Today, we will go through a short history of bioproducts, but we will first start with basic microbial introduction. Even though I believe you all know, I would really like to refresh your memories. So, when we talk about microbes, what are we considering microbes are? So these organisms, microbes, are defined as organisms, acellular or basically microorganisms are defined as organisms and acellular biological entities that are too small to be seen by naked eye. Now, the size is not the only feature that defines them. So it's not just because it's micro, it's a small organism, and that's why it's microorganism. There are other features that define microorganisms, and that would be definition of and organization of cellular structure. But in summary, we usually say that every organism that is smaller in diameter than 1 millimeter so it can be unicellular. It can be unicellular, and if it's multicellular, it just must lack differentiated tissues. So organism cannot be considered prokaryote, actually microbe, if it has tissues. And that, I believe those are all logical things that you already know. Now, broadly, we can divide microbes into cellular and acellular forms, and those acellular forms are viruses, prions, and virusoids, and cellular forms are fungi, protists, and bacteria, and of course, archaea. And in this class, we will talk mostly about bacteria and some archaea, but mostly bacteria. I will mention a few archaeas, but in applied microbiology, archaea are not very useful. They can be interested from some points, but not from points of applied microbiology. Now, so both bacteria and archaea are defined as 2 different branches in phylogenetic tree. So what you see here is a standard image of phylogenetic tree that is based on 16S ribosomal RNA sequencing, and you probably all know that early divergence resulted in bacteria and archaea, and then eukaryotes were separate line. But my point here, what I'm trying to show you with this picture, is that even though we know there are 3 kingdoms, 3 domains of life, bacteria and archaea belong to prokaryotes. Eukaryotes are separate lineage, and if you look in details, eukaryotes, or humans and animals, especially humans and animals, are only one small portion of this phylogenetic tree, of this life. So basically, the world is dominated by microbes, is what I'm trying to say here. My former boss used to like to say how this planet and us are nothing but a big sandwich that microbes feed on. And if you think about that, it's kind of true. Microbes live anywhere. They live on us. So they're We are not superior creatures. They are. And this is what I'm trying to say here. So basically, bacteria and archaea, again, especially bacteria, for this class, but both of those type of organisms live in many extreme environments. And for this class, is very important to understand those extreme microbiology, because very often the feature of organisms that can live at hot, cold, salty stress, acidic stress can be used in biotechnology to create some proper product or enzyme, or to detect certain gene that can be implemented in different organisms. So it's kind of important for bioprospective of different types of materials, genes, organisms, proteins, but enzymes are proteins. So yeah, anyway, even though we say that single microbial cell is much smaller in diameter than a eukaryotic cell, the microbial cell itself is very complex structure, and there are certain features that distinguish it from eukaryotic cells, and should be just a recap of everything what you already know. So basically, in addition, complex and very unique microbial physiology enables them to live in those extreme environments where neither of us would be able to survive. So basically, microbes evolved to utilize anything available in terms of carbon and nitrogen source, and we can use those features to develop many biotechnologies, which we will talk about in this class. Basically, if you know all of this, can you really think that we are superior species comparing to microbes? I simply don't think that. Just think about that. Neither of us has enzymes that can operate at 120 degrees, but we wouldn't be able to do PCRs with Taq polymerase if we didn't find that enzyme, and where we found it, in microbes. So anyway, back to the microbial cell. So in terms of differences between microbial, between prokaryotic and eukaryotic cell, there's certain features that are shared between prokaryotes and eukaryotes. For example, both of them have DNA and RNA present. Both of them have cell membrane and ribosomes, but the key difference is in the DNA organization and structure, and of course in the presence of organelles in eukaryotes, which prokaryotes do not have. So they do not have organized membranes, there is no nucleus, DNA is packed in a nucleoid, so it's a circular DNA. They have a lot of, like many times, you will see a lot of plasmids, which are extra chromosomal materials, and plasmids are very significant for synthetic biology, which we will also cover in certain portion of this class. Now, in terms of applied or industrial microbiology and biotechnology, so the purpose of this science, so to say, is basically put the microbe into the practical use. So make a use of microbes. Even though I love microbes, just being who they are, many times you will have a hard time explaining to people why you are studying certain microbe, unless that microbe has some potential benefits in terms of industrial production. And lucky for us, they do have it, many of them. And there are endless sources of different microbes that can be later used. But again, back to the lecture. Realistically, profit is what matters. And this is something that I don't like, but apparently it is really important. Whatever you want to do with a microbe, it has to pay off. So it has to be cost effective. And again, just because something can be done doesn't mean that it will be done in industry. So it must be market-driven reason. And in your presentations, you will have to have that justification. So again, back to my research that I'm mentioning in previous class. Even though I love making, I think it would be fantastic to make polyhydroxybutyrate from cheap carbon source like methane. And there's technically, it's not cost effective. And we are having a hard time putting that into the large production. There are large productions, but we still use regular plastic. We still didn't completely remove it. We still use fossil fuels. We didn't move to the biofuels, and most likely that's because it's not enough cost effective. So that's a component that you have to consider. Now, that's why this is a competition with the chemical industry. So why would that? So why would you compete with? So what that competition with chemical industry means? So how will you justify it? So basically, microbes maybe can make cheaper chemicals. So that's how you can justify it. Again, it depends on the chemical. So maybe with microbe it will be cheaper. What will that you can always use as justification when you compete with chemical industry is that microbes make cleaner chemicals. So it's called green biotechnology. However, sometimes you have to use harmful reagents to extract products. So that's kind of counterproductive. But then you can explain that like you can have alternative approach to extract products that does not involve toxic solvents. So microbes may make more specific chemicals, which goes in your favor, in our favor, and also more complex chemicals. So again, this course has different perspectives than most theoretical microbial courses, so I am not trying to bother you too much with the theory. I'm trying to teach you how to think if you are about to make your company. So you need to understand how to control and explore microbial activity, especially microbial metabolism. That is very important, but you also have to understand current situation in market and in the world, and to consider both pros and cons. So a little bit about the history. I mean, there are many things to say, but I try to condense it so you can get a big sense of what was happening. Basically, until the late 1800s, microbes were used to make food and beverage. And this is something that you probably also read about. So Babylonians were making beer. Middle East, people from Middle East made wine. Egyptians made bread. There was a famous pickling vegetables. In my village, some people still do that just for fun. Fermenting milk still does it. Pickling vegetables is actually done commercially, just some people like to do it at home. It tastes way better. Before 1670, microbes were used just empirically. So before all these things were starting to happen, microbes were not just empirical. So when they were first seen in 1670 by Leeuwenhoek, he called them animalcules. So that's a archaic term for microscopic animals. So after that, the expansion started. Well, the expansion, yes. So basically, only two industrial fermentations were known before 1800s. So that was ethanol, wine and beer, and vinegar. Both of them are kind of important these days. First commercial brewery appeared in 1700. So, and there is a nice picture here, ancient picture. And these breweries actually contain wooden vats up to 75,000 liters. Fantastic amount of alcohol. So those wooden vats were occasionally lined with copper, but regardless of copper lines, they had really a problem with the cleanliness. Louis Pasteur, who was living in mid to late 800s. He developed the concept of pasteurization. Basically, that would be a heat treatment to decrease spoilage of organisms. Again, a little bit more what happened after, actually in late 800s. So in 1860, Pasteur showed that microbes can be obligatory fermenters. So by the end of the era, they had pure starter fermentative culture. So pretty early beer was made, right? And then in the beginning of 1800s, there was a lactic acid fermentation. Little before that was ethanol industry. So again, Carlsberg's beer, but basically some basic levels of fermentation, lactic acid, vinegar, ethanol. So those things maybe look basic today, but they are kind of significant for everything else that we now do. Now, in some modern times, in between 900 and 940s, there were some new food-related and chemical products. So basically, there was production of baker's yeast for baking and brewing. You all probably heard about that. Lactic acid, citric acid for food industry, glycerol, acetone, butanol used in military services. There was an ethanol for fuel, and there is a picture of this cool Ford Model T that ran on ethanol. You can Google a little bit of that. There are a few cool articles. organic acids for plastic synthesis, petrochemistry. So basically, this is the beginning of competition between microbes and fossil fuels for feedstocks. So a few interesting facts about military demand that drives fermentation in between this period, 900 to 940s. So what happened in the World War I, these are just years, of course, I don't expect you to memorize those years. So in World War I, glycerol was needed to make explosives, nitroglycerin, right? So Great Britain blocked Germany, and Germany lost supplies of glycerol feedstock to make nitroglycerin or explosive. Now, yeast was treated with sodium bisulfate to produce glycerol, and then Germany converted to microbial production of glycerol by yeast. So again, there are many articles about that you can read. This is just a brief information that I wanted you to figure out, so that there was a constant need of certain carbon sources. So like in World War I, there was a need for glycerol. So Germans started to make glycerol from yeast. So they even then they knew that it can be very useful. So for World War II, there was the use of this bacteria Clostridium acetobutylicum. That's a famous organism that we will talk about later in class. So this organism was used to produce various products that could be used as a weapon. So some of them are listed here. So acetone and butanol through fermentation. So butanol was used to make a rubber. Rubber can be used for tires. Acetone can be used to make another type of explosive. So various things can be made from microbes that are not very nice, but useful for certain people. And microbes, as I said, microbes are involved in everything, including politics. So basically, there is this Chaim Weizmann and Balfour Declaration and the State of Israel. So Weizmann's fermentation process from cheap starch was a key to produce explosives and tires in world wars. So they needed butanol for rubber, as we previously said. Acetone was needed for cordial. So that kind of drag attention of Churchill, Winston Churchill. So UK Foreign Secretary signed this declaration, which proclaimed British support of Jewish state in 1917 to reward Weizmann for its war effort. So again, fun fact. You can read more if you are interested in history part. So in World War II, there was the beginning of this modern bioterrorism. So anthrax was produced, and after 1940, solvent fermentation started driving technological advances. So what does that mean? So we could see, after 1940s, we could see those stainless steel fermentation vessels. Some of them we will see also when we visit the Bressler lab, just very modern technologies, not those ancient ones. So those vessels must be clean, but because they're stainless, they're steel, they can be sterilized, and you can have temperature control, which is a big issue, so you can control the process. After the 1940s, pure cultures were used as inoculum, although nowadays we often talk about using microbial communities, so pure culture does not always have to be the best choice, which you will see in class later. Basically, there were some examples of starting with a pure culture and sterile media, so that was the beginning. Then fermentation was established, and when this establishing fermentation created condition that can maintain pure culture. So again, controlled process, everything must be clean and sterile. In this period also, inoculum train was developed, and we will learn in details about inoculum train later in class. But I'm just giving you here a brief overview that basically you have to have aseptic, sterile conditions during inoculum preparation, because you have to maintain that sterility. And you have large-scale anaerobic culture, and in order to inoculate, you start from a smaller and you go to the large. So you don't inoculate 5 mils into the 500 500 liters reactor. So you have to start with an inoculum train. You go from small and then you go up. Again, we'll talk in details in one of next class. So after 1940, that was antibiotic period. So there was a need for penicillin antibiotic in Second World War. So, and there was demand for large-scale fermentation of this antibiotic, which is made by filamentous fungus that is shown here on picture. So initial cultivation of Penicillium fungus was in surface culture in steel bed pans. So that is some sort of fermenter. Well, not really fermenter, but it is bioreactor. The production, though, was limited in volume. But antibiotic demand actually drives technology advances. So antibiotics were highly significant after 1940s, and they still are. They save many lives, that's for sure, although we do have to be careful with the usage of them so we avoid spreading of antibiotic resistance that can be transferred to toxic pathogens. But anyway, back to the antibiotic demands that leads technology advances. So people start paying attention to the strain improvement. Basically, there was this penicillium strain isolated from cantaloupes that produced penicillin in submerged culture. So they did mutations and selections to improve yields like 2,300 times. So that was highly significant. So that was like strain improvement. Then people paid more attention on aseptic conditions. So basically, clean but not sterile or sterile enough conditions are not good enough to have protective fermentations, because what I'm trying to say is that initially people were more focused on this. Yes, it's sterile enough approach, but if you want to have protective fermentation, then it has to be completely sterile. So even though somebody can tell, oh, maybe probability of contamination is low, that's still not enough. Like, you shouldn't take that risk or you can lose production. Now, unless you're working with mixed culture, but even then. Then people started developing those antibiotic producers, those slow-growing fungi, and they basically, all those slow-growing fungi and vaccine production, they all require completely sterile media. So if you want to make something that will be used by people, it cannot be contaminated with random pathogens. So this need for antibiotics and for vaccines would require protective fermentation. That's why people start paying attention more to aseptic techniques. So sterile enough was not good enough anymore. But again, these measures were very complicated and extensive, and that would, as you can guess, would be very expensive. So those extensive measures were used exclusively, as I said, for those products like antibiotics, amino acids, vaccines, something that will be consumed by people or animals. Then a few more fun facts. So there are, even though I said that aseptic techniques are important, so when we talk about public health, municipal waste, and the environment, there are some non-aseptic biotechnologies like wastewater treatment, municipal composting. So wastewater treatment and composting are two largest and least recognized fermentation capacities worldwide. And this fermentation, but it's non-septic, so it's non-aseptic, so it's not sterile. Then bioremediation, so basically when you treat a soil or water with some microbes that can collect toxic chemicals from that environment and ingest it, so it doesn't have to be sterile. Bioleaching as a means of mining of low-grade ores, so basically you use microbes to extract metals, especially iron, copper, and gold. That's a very famous technology used in mining industry. Then in 60s, so microbial biomass was being used as a source of proteins to supplement animal fat. So you would see this single cell protein, SCP. Basically, those are you can also use whole cells with high protein content. So if you have a cell that generates like 70% of dry weight of protein, so for example, yeast can grow on petroleum and it makes a lot of protein, and then you can use that as a fed usually for some small animals like salmon or chicken broilers. Then this bacterium Methylophilus methylotrophus can grow on methane and make this protein. So it's a single cell protein made from methane. So if we talk about methane oxidizing organisms, this will be one of examples of value-added products that can be made from methane. And I will talk about, we will have a lecture about C1 metabolism. So basically this was like $100 million investment, but because of soy and fish meal, who were cheaper, this didn't really develop well. So again, you always have to think about what your market requires. Current single cell products, food products, single cell proteins. So food for humans is this corn made from Fusarium venetatum. So don't bother memorizing the fungal name. It's filamentous fungus, and it is using starch as a carbon source. So it's available in US and UK, not in Canada. And there is this health food supplement, Cyanobacterium spirulina, again, approved for human use. Now, in 70s and 80s, Cohen and Boyer discovered restriction enzymes. So this is revolution for synthetic biology. So these enzymes enable cloning, but of course you will run into the social issues. GMO versus non-GMO. Is it ethical or not? Should you use GMO to increase food supplies or not? That's debate, and it can be highly extensive. So we have this progress in development of recombinant DNA. And when DNA sequencing technology developed, this was a big boom and big revolution in every field, pretty much in not just in industry, but also in any I think in any type of scientific research, like not just in microbiology, but pretty much whichever field of science, I mean biological science, you are exploring, sequencing is something that you will use for sure. Previously, sequencing was done manually. I heard stories, I never done it, but I know that people were doing it, and I heard it's extremely expensive. Today, that's automatized process. So you can get a sequence of whole genome in a few hours, depending on when you send it. In 80s, PCR was developed, like polymerase chain reaction, another revolutionary technology. So that enables screening, sequencing, control mutation, basically everything what you can think of when you say synthetic biology. So in 70s and 80s, again, there were way more sophisticated applications. Monoclonal antibodies, recombinant proteins, GMO crops. So these monoclonal antibodies are basic diagnostic tests. There are several treatments for cancer that include monoclonal antibodies. In terms of recombinant protein production, you probably all heard about human insulin that was expressed in E. coli, then growth hormone, clotting factor. There are various hormones that have been produced, like recombinant proteins. Genetically modified organisms, so canola, this is Roundup Ready. So this canola has glyphosate resistance gene for salmonella, GMO corn and cotton with protoxin gene from bacillus. So these crops have been developed after technologies were established, like PCR, first restriction enzymes, then sequencing, then PCR. So, and that was a revolution. Now, in 21st century, the beauty, biofuels are continuously developing, and we will talk in this class about first, second, third, and next generation of biofuels. So, first generation of biofuel includes ethanol made from cornstarch and sugar cane. Then little progress was made with the second generation, where also ethanol was made, but this time from lignocellulose. Then we will talk about these different sources for biofuel design. that kind of matters a lot. And the third generation of biofuels involves biofuels produced from algae. So this is kind of fantastic because yeah, we'll talk about that. I'm not going to do spoilers. And last but not least, the fourth generation of biofuels includes the use of synthetic biology and GMOs. I'm personally the biggest fan of third generation biofuels and fourth, of course, because I love synthetic biology. So speaking about synthetic biology, it is a platform for chemicals and polymer synthesis. It cannot always be done, so you have to be very careful on what are you choosing and when are you choosing. As I said at the beginning, just because something can be made doesn't mean it will be, it will give results when you do it. So there are many other considerations. So we will at the end talk about these integrated biorefineries. So basically, the goal is to have minimum waste and maximum diversification of product streams. So get as much product as you can, don't waste anything. That will be basically the closure in this class. Integrated biorefineries. How can you close the loop, make a sustainable solution? And when you make your own projects, that can be one of your topics. So saving the environment is a big deal, and this is something on what we all have to work. So maybe neither of us individually can do much. Like I would say very often, well, I cannot do much as individual, but I don't say that anymore, because if each of us does what we can, so we just basically add a small piece to the basket, I'm sure we can make this planet a better place. And we do need that. So this is something that you all want to know. What would you have to know from all these interesting historical facts? Now, you have to know about some basic social, political, and technological circumstances that have driven development of biotechnology. So obviously, I won't ask you to list me years when some revolution happened in biotechnology, but I would like you to understand that increasing population size increased the need for increased food production, and then that fear of food shortage always was present, especially in third world countries. Then the development and the progress of wars, both wars, first and second, transportation and industry increases the need for chemical feedstock. So think about that need for butanol, for glycerol, acetone. So all those are military requirements. Now, public health concerns exist related to the need for antibiotics, vaccines, and monoclonal antibodies. So you need to improve human health, and as we mentioned, many products like monoclonal antibodies are used in cancer treatments. Manufacturing and synthesis can bring up the question of management of waste, and management of waste is something that you have to consider whatever you are making. So when you make your mini company, waste management will be one of portions in your web page. You have to describe what will you do with the waste. So in ideal world, you would have that sustainable loop, and you can sell that waste to some company that can make a use of that waste. So again, I'm just giving you hints. Anyway, so this is all about social issues. So basically just get the general concept. Then gradual progression and integration of techniques and components of fermentation and microbial technology. So what you need to know is that applied microbiology did not start with cloning. So it does not always require cloning. So I know that many people, whenever we say applied microbiology, will say, oh, synbio, yes, and it's great. And sometimes it's fantastic, but sometimes there is no need for that. Sometimes cloning can help, but not always. Sometimes cloning is just not needed. Now, it can be useful, especially when you need high volume of production, so when you want to scale something up. But even when you want to improve production with a cloning, like you overexpress or you mutate gene or you change the gene, there are other factors that you have to consider in terms of metabolism. So again, cloning does not always have to be the best solution. And it is useful for modification of processes, but again, not always the case. For applied microbiology, it's extremely important to have to be accepted by society, and society must be aware of all possible potentials of microbes in the world, which is what I'm hoping we all are doing. And basically, the way how I want to end this is be informed citizen. Now, there were those, and yeah, I had a few case studies that I wanted to share with you, and this is why it's actually important to be informed citizen. So again, the quality of this website is not very certain, so I'm not going to, I just took information from there. So this was the case study with genetically modified tryptophan. Tryptophan is amino acid. So everything started in late 80s, 1980s. So this amino acid, L-tryptophan, is essential amino acid, and it's sold globally as a supplement to the diet. So this is not a drug, so it's basically a dietary supplement. So because it's not a drug, the dose doesn't have to be specified. So this is not a medicine, it's just food supplement. Now, so what happened is that some people consumed a lot, apparently more than 4 grams per day. Again, the dose was not present, so they just didn't know. So this Japanese company, Showa Denko, made this tryptophan using bacillus, and at some point they switched to GMO bacillus. So Denko then downgrades purification methods, and that affected downstream processing. Now, before spring 1989, many consumers, like more than 5,000, so people who consumed this L-tryptophan from genetically modified organism, became very ill. Later on, around 100 of them died from some weird disease. And there is an article here. So the only link between patients was this L-tryptophan that Showa Denko made. So that was the only connection. Again, there are no clear conclusions on this website. So eventually, some rigorous analysis of the product revealed that there were over 60% impurities, including 0.1% of tryptophan dimer, and in this case, genetic engineering was blamed for toxic effect. You could imagine what happened, how people were pissed about genetic engineering then. So what the company could do only to withdraw the product. After they withdrew it, the number of cases of field people decreased. So surprised. So Showa Denko destroyed all GMO species, so everything what they made, all bacillus organisms, but they had to pay out a lot of money for more than 2,000 claimants. Obviously, you don't have to memorize those numbers, but this is a good example of how genetic engineering didn't really increase it. Honestly, I love cloning, and I would clone whatever I can, but I learned this from people who are more experienced than I am. And sometimes really cloning is not a solution. And when I come back from this conference, I will share with you some ideas that I gain and something that I learned when you don't actually have to do cloning, and sometimes cloning is not even beneficial. Now, what happened with this Showa Denko? Apparently nobody knows. We can guess. Strain doesn't exist, so we cannot do any modifications. So they made a very smart move. And there are possible explanations, just paraphrased. I paraphrased from this web page. So maybe that inserted gene had higher expression level that resulted in overproduction of L-tryptophan. So that literally caused the overdosing. High concentrations high concentrations may have created conditions conductive to spontaneous or enzymatic dimerization. So all L-tryptophan might be transformed by some unidentified enzymatic reactions. This inserted gene that resulted in overexpression made most sense to me. And the third one is that high tryptophan concentration might be toxic to the producing strain, inducing mutations, or basically selecting for strains that form side products. So first and third explanation made most sense to me, that when they did modification, the inserted gene messed up something in genome that resulted in overproduction of tryptophan. Maybe what would happen, they maybe purposefully wanted to have more tryptophan, but then so much tryptophan was toxic to the producing strain, and that toxicity caused mutations. So again, we will unfortunately never find out. So this will be all that I had to share with you for this class. I hope this was fun enough, not many things to memorize. And we will go through review session before the exam, that's for sure. I will stop recording now, and I'm looking forward to seeing you all on Tuesday when I come back. You guys will be the first class that I will see at 9:30. I hope I arrive safe, and have a great rest of the weekend. See you soon.